Hello students, today I am going to give brief introduction about the structure of gluten protein and the role of gluten in bread making. Gluten is defined as a protein fraction from wheat, rye, barley, oats or their crossbred varieties and derivatives thereof, which is insoluble in water. Gluten proteins are storage proteins and occur excessively in the starchy endosperm of the grains and make up around 70 to 80% of total grain proteins. The remaining grain proteins are water and salt-soluble albumins and globulins, mainly enzymes and enzyme inhibitors and insoluble structural proteins, such as membrane and lipoproteins. Traditionally, gluten proteins have been divided into two fractions based on the difference in solubility, prolamins and glutenins. The prolamin fraction contains mainly monomeric proteins which are insoluble in water and salt solutions, but soluble in aqueous alcohol, that is 60% ethanol or 50% propanol, whereas glutenins are polymerized by interchange disulfide bonds and are insoluble in water, salt and aqueous alcohol solutions. They are only partly soluble in dilute acids and bases or solvents containing detergents. Wheat prolamins are called glidins and glutenins. Barley prolamins are hodins. Rice prolamins are scalins. Oat prolamins are avenins. Maize prolamins are zein. Rice prolamins are oryzins. And kefirins are the prolamins of millet and sorghum. Glidin and glutenin are known to impart entirely different physical properties to the gluten network in dough. Glidin behaves mainly as a viscous liquid when hydrated and imparts extensibility, allowing the dough to rise during fermentation, whereas glutenin provides elasticity and strength, preventing the dough from being overextended and collapsing, either during fermentation or in baking. Classification and structural features of gluten protein. According to Osborne fractionation, 1907, grain proteins on the basis of their solubility can be divided into albumins, that is water soluble, globulins, that is salt soluble, prolamins, that is alcohol soluble, and glutenins, that are alkali soluble. Albumins and globulins contain higher quantity of tryptophan and arginine, whereas glidins and glutenins are rich in glutamine, proline, and phenylalanine. Amino acid sequencing indicates that the storage proteins of wheat, rye, barley and oats are closely related and differ from those of other cereals. Based on homologous sequence and similar molecular weights, they can be divided into three groups. First, the high molecular weight group, second, the medium molecular weight group, and the third, the lower molecular weight group. Shivery and colleagues have proposed an alternative classification in 1986 that reflect biological, chemical and genetic relationship among components polypeptides of the gluten complex. They divided gluten proteins into three main categories, namely sulfur-poor prolamins, sulfur-rich prolamins and high molecular weight prolamins. All these gluten protein groups are considered typical prolamins because they are rich in proline and glutamine amino acid residues and are extractable at least partially in aqueous alcohol, particularly after addition of reducing agent. The high molecular weight prolamins contain three protein types: high molecular weight glutenin subunits of wheat, high molecular weight scalins of rye, and D-hodins of barley. Oat proteins are missing from this group. Proteins of the high molecular weight group consists of around 600 to 800 amino acid residues corresponding to molecular weights of around 63,000 to 88,000. The amino acid compositions are characterized by high content of glutamine, glycine and proline, which account for about 60% of the total amino acid content. The high molecular weight subunits are further divided into X-type and Y-type. The X-type is somewhat larger polypeptides, molecular weight 83,000 to 88,000, while as the Y-type is smaller, molecular weight between 67,000 to 74,000. DNA sequencing technique revealed that all types of gluten protein polypeptides have at least 2 or 3 distinct structural domains. A central repetitive domain flanked by non-repetitive C-terminal and N-terminal domains. High molecular weight prolomins have the majority of their cysteine residues in the N-terminal domains and only one in the C-terminal domain. Several Y-type subunits have one additional residue in the central repetitive domain towards the C-terminal end. An X-type subunit has one residue close to the N-terminal end of its central domain. The medium molecular weight group, which is sulfur-poor prolomins, consists of the homologous omega-1,2 glidins of wheat, omega-scalins of rye, C-hodin of barley. These proteins consist of 320 to 380 amino acid residues, which correspond to molecular weights of around 40,000. Besides omega-1,2 glidin, wheat contains unique omega-5 glidins with around 420 residues and molecular weights of approximately 50,000. These type of proteins are also absent in oats. Medium molecular weight group contain high content of glutamine, proline and phenylalanine, which together account for about 80% of total amino acid residues. Sulfur-poor prolomins have no cysteine residue. The low molecular weight group, that is sulfur-rich prolomins, contain monomeric proteins including gamma and gamma glidins, gamma 40K scalin, gamma hodins and avenins. Their sequence consists of approximately 300 amino acid residues corresponding to molecular weights of around 28,000 to 35,000. The amino acid composition is dominated by glutamine and proline and high content of hydrophobic amino acids such as leucine and valine. The sulfur-rich prolomins are characterized by a shorter repetitive domain and a longer C-terminal domain compared with high molecular weight prolomins and sulfur-poor prolomins. Sulfur-rich prolomins have their cysteine residues in the C-terminal domain, the only exception being the low molecular weight subunits, which also contain one cysteine residue in the N-terminal domain. Total numbers and relative position of cysteine residues are important for polymer size and the different polymerization behavior of glidins and glutenin subunits. The presence of cysteine residues in the form of intramolecular disulfide bonds in glidin and intramolecular disulfide bond in glutenin subunits is due to the fact that glidins have even number of cysteine residues, whereas gluten subunits have odd number of residues within a single structural domain. It is the intermolecular disulfide linkages which are considered responsible for the giant size of glutenin polymer. It has been suggested that intermolecular sulfur-sulfur bonds are limited. That is only 2 per polypeptide chain connecting each chain in a head-to-tail fashion to the next chain, thus forming a linear macropolymer of glutenin known as concatenations. Bonds involved in the glidin-glutenin interaction. During the early stage of dough mixing, glutenin interacts with glidin to form gluten, the viscoelastic matrix of the dough. Several chemical bonds are involved in the development of the optimum dough structure. Disulfide bonds are the principal covalent bonds within and between gluten polypeptides. Non-covalent interactions include ionic, hydrogen and van der Waals interactions. Glidins have either intramolecular and disulfide linkages, whereas in glutenin there are both inter- and intramolecular disulfide linkages. Disulfide bonds play a key role in the formation and development of dough. They form strong cross-links within and between polypeptide chains, thereby stabilizing hydrogen bonds and hydrophobic interactions. During dough formation and development, disulfide bonds can be mobilized through disulfide interchange reactions. The interchange reaction requires a sulfahydryl containing substance to initiate the series of disulfide interchanges resulting in extensive polymerization of the gluten proteins. The total number of sulfur-sulfur bonds do not change, only their location in the glutenin molecule is altered. Hydrogen bonds are formed as a result of the affinity of hydrogen atoms in hydroxyl, amide or carboxyl groups for oxygen in carboxyl or carbonyl groups. Gluten proteins are rich in highly polar amino acids, in particular glutamine. Glutamine constitutes over 33% of the amino acids present in the gluten. The amide group in glutamine actively participates in hydrogen bonding, and in fact it forms 2 hydrogen bonds per glutamine residue. Hydrogen bonds are much weaker than gluten bonds, but because of their large number that act cooperatively and contribute significantly to the structure of the dough. Another unique feature of hydrogen bonds is their ability to interchange under stress and thereby facilitate reorientation of gluten proteins. Gluten proteins also contain large proportion of apolar amino acids such as phenylalanine and proline, which are considered potential source of hydrophobic interactions. Their functionality is similar to that of hydrogen bonds, but the overall effect is much smaller. Hydrophobic interactions differ from other chemical bonds because their energy increases with increasing temperature, which could result in increased stability during baking. Another important feature of gluten proteins is that they have a very low charge density. This is due to their low level of basic amino acids such as lysine, histidine, arginine and tryptophan, and also due to the fact that glutamic and aspartic acids occur mainly as amides. As a consequence of this low charge density, the wheat gluten proteins are not repelled and associate strongly by non-covalent interactions. Such behavior is important to baking technology in that it results in the ability of gluten proteins to form viscoelastic gluten network that is essential for gas retention. The linear glutenin hypothesis proposed by Evert has received wide acceptance in explaining the viscoelasticity of gluten. According to Evert, the glutenin polypeptide chains from long linear concatenations with 2 sulfur-sulfur bonds connecting each chain in a head-to-tail fashion to the next chain. The entanglement may be purely physical, and at the point of entanglement known as nodes, non-covalent interactions might occur to form crosslinks. Under stress conditions, the entanglement and non-covalently cross-linked structure in glutenin offers resistance to deformation, which is manifested in increased elasticity. It also enables glutenin polymer to recoil after the stress conditions are withdrawn. Viscous flow depends predominantly on molecular spillage at nodes and liable nature of weak secondary forces acting between glutenin polymers. Sulfahydryl disulfide bonds interchange and mechanical scission of sulfur-sulfur bonds may also contribute to viscous flow. Mechanical scission of sulfur-sulfur bonds occurs when the rate of deformation exceeds the rate at which molecular spillage or sulfur-hydrogen sulfur-sulfur interchange can occur, resulting in glutenin polymers being subjected to stress values more than their elastic limits. Gluten in bread making. Bread making process is accomplished through 3 basic operations, that is mixing, fermentation and baking. Mixing transforms the flour and water into cohesive viscoelastic dough. Another key function of the mixing operation is the incorporation of air. Air is essential for introduction of gas cells into which the carbon dioxide produced by the yeasts during fermentation diffuses, as yeasts cannot produce new gas cells. If there is no air cell, the crumb of the final bread would be coarse with few large cells. Several physical and chemical transformation occurs during mixing and kneading of a mixture of flour and water. Under the applied shear and tensile forces, gluten proteins absorb water and partially unfold. The partial unfolding of protein molecules facilitates hydrophobic interactions and sulfhydryl disulfide interchange reactions, which result in formation of thread-like polymers. These linear polymers in turn are believed to interact with each other via hydrogen bonding, hydrophobic associations and disulfide cross-linking to form a sheet-like film capable of entrapping gas. Because of these transformations in gluten, the resistance of the dough increases with time until a maximum is reached, and this is followed by a decrease in resistance indicative of a breakdown in the network structure. The breakdown involves annihilation of polymers in the direction of shear and some scission of disulfide crosslinks, which reduce the polymer size. The time it takes to reach maximum dough strength during kneading is used as a measure of wheat quality for bread making, a longer time indicating better quality. The viscoelasticity of dough is related to the extent of sulfhydryl disulfide interchange reactions. This is supported by the fact that when reducing agents such as cysteine or sulfhydryl blocking agents such as N-ethyl maleimide are added to dough, viscoelasticity decreases greatly. On the other hand, addition of oxidizing agents such as iodates and bromates increase the elasticity of the dough. This implies that the gluten rich in SH and SS bonds might possess superior bread making qualities. Yeasts ferment sugars and continuously produce carbon dioxide in the aqueous dough phase. When the aqueous dough phase is saturated with carbon dioxide, most of the carbon dioxide diffuses into the air cells that are formed in the dough during mixing. The diffusion of carbon dioxide into gas cells increases the pressure within the gas cells that produce the driving force for dough expansion. The viscous flow properties of dough, owing to its monomeric proteins, mainly gliadins, allow the gas cells to expand and release the pressure. During baking, the temperature increases, water evaporates from the liquid dough phase into the gas cells. Carbon dioxide and ethanol produced by yeast also diffuses into the gas cells, resulting in overall increase in the pressure within the gas cells. The viscous component, that is monomeric proteins, mainly gliadins, allow gas cells to expand to equalize the internal gas cell pressure, whereas the elastic component, that is polymeric proteins, mainly glutenins, provide strength to prevent gas cells from over-expanding and thus preventing the rupture of gluten network enveloping the gas cell. Factors affecting bread making quality. Protein quantity and quality. It has long been established that bread making performance of wheat flours are related to the quantity and quality of their proteins. When the total protein content of a wheat grain increases, the total amount of gluten proteins also increases. But the amount of non-gluten forming proteins, that is albumins and globulins, changes very little. Thus, wheat of high protein content usually has a higher proportion of gluten proteins compared with those with lower protein contents. Fractionation and reconstitution studies, that is experiments in which the different flour constituents are first separated and then systematically exchanged between reconstituted flours of good and poor quality, have confirmed that the gluten proteins are primarily responsible for quality differences among wheat varieties. Gliadin and glutenin ratio. Difference in the gliadin to glutenin ratio among wheat cultivars have long been considered an important source of inter-cultivar variation in physical properties and bread making quality. The technological significance of gliadin and glutenin in bread making has been attributed to their contribution to dough extensibility and elasticity respectively. It has been reported that the addition of complete gliadin and its subgroups substantially improved bread making quality as measured by loaf volume. Molecular size distribution. Molecular size distribution has been considered another important factor in relation to functional properties of flour. Gel filtration chromatography studies have indicated that the glutenin fractions of flours with longer mixing times have a higher average molecular weight than the glutenin fractions of short mixing flours. Flours with short mixing time give doughs that break down rapidly during mixing and are easily extensible. Such flours are considered unsuitable for bread making. Conversely, flours with longer mixing times are noted for doughs of high mixing stability and great resistance to extension, which performs well in bread making. Glutenin polypeptides. Up to 20 different high molecular weight subunits have been identified in different bread wheats. Each bread wheat cultivar contains 3 to 5 high molecular weight subunits, which together account for about 1% of the dry weight of the mature endosperm of a wheat grain. This indicates that although high molecular weight subunits are quantitatively minor, but they are functionally important polypeptides of gluten proteins. The high molecular weight subunits of glutenin are encoded at the GLU1 loci on the long arms of the chromosome 1A, 1B and 1D. These loci are designated as GLU-A1, GLU-B1 and GLU-D1 respectively. GLU1 quality scores are assigned to the individual subunit or subunit pair based on their value determined by the SDS segmentation test, an indirect measure of bread making quality. The GLU1 scores of bread wheats usually range from 3 for poor bread making quality to 10 for good bread making quality. This was all about the role of gluten in bread making. Hope all of you have understood. Thank you so much.